Welcome to Chem 313, Lecture 5. To review from the first week of material, last week we introduced protein structure, and we said that protein structure works at a variety of different levels. We can talk about what we call the primary structure of a protein, which refers to its amino acid sequence. And then we have different levels of structure on top of that. We have secondary structure, which refers to the backbone hydrogen bonding pattern. And we said that there were 4 different types of that I care about you knowing: alpha helix, beta sheet, turn, and coil. We said there is tertiary structure. Tertiary structure has to do with the side chain interactions with one another and with the water, which is our solvent. And then there was quaternary structure, which is the same thing as tertiary structure, but between chains. If you're in a multimeric protein that has more than one polypeptide chain, the chains can bind to one another, and that's referred to as quaternary structure. So not all proteins have quaternary structure. One important point to distinguish is when I say structure, I don't mean structure in the organic chemistry sense of the term. So that is primary structure, right? Primary structure is the chemical structure of our protein molecule. That's what atoms are actually bonded to what atoms. But generally speaking, when we talk about protein structure, what we're really talking about is conformation. So what is the arrangement of everything in three-dimensional space, not necessarily what's connected to what. And as we're going to see today, the conformation of a protein is what determines its function. So I think an important point to note that we're going to come back to again today is that the structure of a protein is a sort of optimum. So it's the best possible solution to the problem of minimizing steric repulsion or steric hindrance, minimizing nonpolar amino acids exposure to water, and then maximize favorable side chain interactions. And that can either be with water, if you have polar or charged groups, or with other amino acids in the same chain. So if you were to take the extra bit of time to look at my short lecture, I called it lecture 4.5, where we just looked at some examples of protein structure. One of the things that I hope you took from that lecture, if you watched it, and I encourage you to, is that even though protein structures look very different, they all have some very common themes within them, especially for globular proteins, and most proteins are globular. So for example, if we started off with a protein structure that may have looked something like this, let's say that we've got an alpha helix, and then a turn, and then our protein chain forms, say, a 3-stranded beta sheet, so something like this, and we've got a loop here, and then we've got another alpha helix lying on top. So one thing that you would have noticed is that all of the nonpolar amino acids are going to face the interior. Why is that? Well, that's because of the hydrophobic effect. So your valine and your leucine and things like that, because water cannot hydrogen bond with it, water is going to tend to sort of sequester them into a droplet. And the interior of the protein is sort of like our droplet. And almost all globular proteins have a nonpolar hydrophobic interior. But then on the surface, you're going to have some groups that can hydrogen bond with water, and then some other groups that are actually going to be interacting with one another. So for example, maybe at the ends of one of these helices, we have a positively charged group, and we've got a negatively charged group at the end of the neighboring helix. And so that would be an example of a tertiary structural contact. And that sort of helps to lock our protein in a particular conformation. Because after all, there's lots and lots of different ways that you can squish a protein chain around so that all of the nonpolar stuff is on the inside, but there's only really one final, ultimate, correct structure for every protein, and that's the one that's going to maximize all our favorable side chain intermolecular forces. And so the exposed amino acids on the surface tend to bind to one another. All right, so the topic of today's lecture is going to be an introduction to protein function, but we are not done with structure yet. We have to answer this question of why does structure really matter? Why have we been spending so much time focusing on the structures of proteins when we haven't even talked about what proteins do yet? Well, as it turns out, the structure of a protein, right, the correct structure of a protein, which we call the native structure, is the structure of the protein that is functional. So we already said before that the primary structure, or the amino acid sequence of a protein, determines its three-dimensional shape. But it can also be shown that the three-dimensional shape of a protein determines its function, what it does. Whether it's an enzyme, whether it's a hormone receptor, whether it's a hormone, whatever. And all of this information is contained within the amino acid sequence of the protein itself. So there was a classic experiment around 1960 that demonstrated this. It was called the RNase unfolding experiment. So what's RNase? RNase, that's this enzyme right here. So RNase is an example of an enzyme, which means it acts as a catalyst for chemical reactions. And this is the reaction that RNase catalyzes. It essentially chops up RNA. If you take RNA in the presence of water, it chops it up into nucleotides. Okay? If you have no RNase, or if you have non-functional RNase, RNA is stable. All right, so this reaction doesn't happen to RNA by itself in an appreciable rate. It takes forever to happen on its own. So for all practical purposes, unless you both have RNase and that RNase is in a functional structure and you add it to RNA, you will not get RNA decomposition. So keep that in mind for a second. So what did they do in this classic experiment? What they did is they started off with native RNase. So I'm going to draw the native structure of the protein as something like this. So this is our RNase. They took native RNase and they mixed it with RNA in a test tube and let it sit for a while. And after sitting for a while, what they observed is they no longer had any RNA in the test tube. All they had were free nucleotides. So what that tells us is that our RNase was active, which is what you would expect because after all, that's RNase. That's what it does. All right, so then what did they do? Well, then they took RNase but they added two chemicals to it before adding the RNA. The two substances they added were urea and beta-mercaptoethanol. Urea is this compound here. So what do you notice about this molecule? Well, we've got electronegative atoms out the wazoo, and we got NH groups down here. So this is a very strong hydrogen bonding molecule. And essentially what it does is it breaks hydrogen bonds, which means it's going to break up secondary structure. Beta-mercaptoethanol is this. Because of this SH group right here, this breaks up disulfide bonds. And RNase has 4 separate disulfide bonds in it that are responsible for holding the structure in place. We've got one there, and then 2 kind of overlapping up at the top. So treating RNase with urea and beta-mercaptoethanol ended up forming this sort of unstructured RNase that no longer has its disulfides and no longer has the correct secondary structure. So they took that chemically treated RNase and RNA, put those in a test tube, waited a while. You still had RNA after the experiment was over. That must mean that our protein became inactive. So what is this telling us? This is telling us that unless your protein has the correct three-dimensional shape, it does not have its proper function. But then what's interesting is if they took the incorrectly folded RNase, the chemically treated RNase, and then removed the urea and removed the mercaptoethanol, so that you ended up with the correctly folded protein once again, and you added that to RNA, now it's active again. So removal of urea and mercaptoethanol restores the activity. Why? Because it restored its structure. So there were 2 conclusions from this experiment. The first thing is that protein molecules inherently know their correct structure, right? So like we set up here, when the protein goes from having a certain primary structure to having a certain shape, this is a process that occurs spontaneously. So we don't need other proteins to give this protein its correct structure. It's not like a manufactured particle where it has to be stamped out by a machine. The protein, as soon as it's synthesized, it automatically adopts the correct structure. And this is something that was really remarkable at the time and completely unexpected. It was presumed that there must be some sort of organelle or something in the cell that causes proteins to have the correct shape. The second conclusion from this experiment is that the three-dimensional shape determines the function. So all the information that a protein needs to be functional is built into the amino acid sequence. As long as you can synthesize a polypeptide that has a particular sequence of amino acids, it will fold into a three-dimensional shape that is optimal for that sequence of amino acids, and that three-dimensional shape will give the protein some kind of function. Now, this is kind of a strange observation because it's almost as if we're saying that the protein has its own mind and it knows what it's supposed to do, which of course is ridiculous. And there's a problem out there that's known as the protein folding problem, which is what this slide is supposed to be indicating. And that's the issue of how can a protein molecule just by itself spontaneously find the correct structure? Because remember, after all, if your protein has N amino acids, we said last time that for each amino acids, we have 2 rotatable bonds per amino acid, right? We have the C alpha to C and the C alpha to N bonds. So we have roughly 2N rotatable chemical bonds. And since most proteins have 100 or more amino acids in them, that's going to be several hundred possible rotatable bonds, and each one of those is going to have several possible conformers. So we've got a huge number of possible structures. And yet what ends up happening is that the protein, every molecule of a given sequence always comes to the same structure. So if we say that this final structure here is our native state, in other words, it's the functional state, well, how is it possible that the protein is just going to find this structure just by randomly fluctuating? Well, it doesn't randomly fluctuate. Remember, the native structure is an optimum. We have optimal intermolecular forces in our native state. Another way of putting that is that the native state is at a minimum of free energy, and therefore it's stable. So if you understand that the native state of a protein is the most stable state, then the protein folding problem becomes a bit more intuitive. So the way it works is like this. Whenever you have a protein that is first synthesized, it truly is just all coil. So at the very end of the course, we'll see how protein molecules are synthesized on the ribosome. And when they're first made, they are indeed just sort of a random string of amino acids. Well, what's going to happen is that because we are not at absolute zero, our molecules are in motion and our bonds are going to be rotating a little bit. And what's going to happen is that after a few fluctuations of your structure, you're going to end up forming some native-like structure. We'll say this turn of alpha helix, which is also present in the native protein. Well, because this is native, it's stable. And once it forms, it persists. And now the rest of the molecule keeps on fluctuating in order to sample different structures. And gradually you're going to form more and more stable native-like structures, which are going to persist until finally you get to the native structure. So the problem gets easier as you go. As stable structures form, they tend to stay there, and only the rest of the molecule needs to alter. And finally, after a few cycles of rotating bonds and moving around under thermal energy, the protein is going to settle into its optimal lowest energy conformation. So it's pretty remarkable that protein molecules can do this. The native state can be formed in roughly a second in most proteins. Whereas if you just truly allowed a random search of every possible protein conformation, it would take years and years and years for the protein to just randomly happen upon the right structure. But it happens quickly, and that's because it doesn't happen randomly. Once you form a stable conformation, the stable conformation stays in place, and only the rest of the molecule has to adjust. So what I hope that slide got you to appreciate is the fact that protein structures are dynamic. So remember, weak intermolecular forces are what govern protein structure. Forces that are much weaker than chemical bonds. And because weak forces govern protein structures, all of our structures are only weakly stable. They're at a minimum of free energy, but it's not a very deep minimum of free energy. And therefore, structures are somewhat variable. And most protein molecules can exist in several different but closely related conformations. So to give you an example of that, I looked up this protein on the PDB. This is a monkey virus protease. And I just showed you the backbone trace there in green on the left. So this is a single structure of the protein. What do I mean by a single structure of the protein? I mean, duh, that's one protein, right? What I mean by that is that this is essentially a snapshot. And when we see our protein structures that you look up on the PDB or that you see in your textbook, essentially what we're seeing is a snapshot of an object that is in motion. If you were to take several snapshots of this protein at different points in time, you would end up with something that looks more like what you see on the right. This is the same protein, but it's an overlay of 10 different structures. And as you see here, what we have is 10 different conformations. Right? And they're all pretty similar, but they're not exactly the same, right? So if you look at the secondary structure, it's pretty much the same between all of the different protein structures. What's different is kind of the orientation of everything. So you'll notice that, for example, this small beta sheet up on the top, it's connected by these two loops to the rest of the molecule. And you'll notice that it can kind of flop around a bit, right? So like the dark purple structure, this beta sheet is kind of sticking straight up, whereas in the white structure, it's all the way over at the left. And this loop that connects that small beta sheet to the rest of the molecule, it's also pretty variable. But if you look at the interior of the protein, like this alpha helix and this helix here, all conformations there are basically the same. So this isn't an accident. It's actually very important that proteins can undergo these sorts of conformational changes, most proteins, because an ability to undergo conformational change is critical to the function of most proteins. If proteins were just chiseled out of rock and they never moved and they just sat there as is, most proteins would be rather useless. So we're going to see over the next couple of slides one way in which the conformational change, or the ability of a protein to undergo conformational change, is important for its function. This slide talks about our first foray into protein function, and that is protein or macromolecular binding. So binding is the simplest thing that proteins do. When we talk about binding, what we mean is reversible association with another molecule by weak intermolecular forces. All right. We do not mean bonding. I cannot emphasize that enough. When we mean binding, we mean interaction by weak intermolecular forces. Bonding refers to covalent bonds, and we're not talking about that. So examples of protein macromolecular binding. This protein molecule that we show right here is an antibody. And antibodies, their entire function is they bind to foreign substances that are called antigens. The abbreviation for antibody is capital AB, and the abbreviation for antigen is capital AG. And they form a complex. And the way we indicate the complex is AB colon AG. So the way that antibodies bind to their antigens is, if you look at the molecule like this, it's got kind of a Y-shaped structure. It's actually 4 chains. We showed an example of this in lecture 4. We've got one chain that's kind of teal, one chain that's kind of olive, one chain that's orange, and one chain that's sort of bronzish brown. And the outermost part of these chains is a portion that's called the variable domain. And the variable domain is the part of the molecule that binds to the antigen. Now, if you were to zoom in on the structure of the variable domain as it's bound to the antigen, this is what you would see. We have our antibody molecule on the left, and then in purple is our antigen. And what do you observe? What we observe is that we've got a whole bunch of weak intermolecular forces all along this interface, right? That's between the antibody variable domain and this antigen protein. And the antigen protein, just for fun, I picked up the SARS COVID-19 virus receptor binding protein. So the structure of the virus is known. This is the protein that the immune system seems to be reacting to. All right, so how do these things bind together? Well, they bind together just by forming reversible weak intermolecular forces, the same ones that we've seen before. Hydrogen bonding, the hydrophobic effect, those are the big two. Another example involves not antibodies, but enzymes. So an enzyme, we already said, is a protein that acts as a catalyst, and the reactant that it catalyzes a chemical reaction on is called the substrate, which is a stupid word, but we're stuck with it for historical reasons. And so enzymes and substrates, as we're going to see later in the week, in order for this reaction to occur, they have to bind to one another to form a complex. And so in this case, this figure down at the bottom shows the enzyme glucokinase binding to glucose. That's glucose there, to form a complex. How does it do this? Once again, just through formation of weak, reversible intermolecular forces. All right, so I wanted to illustrate a hypothetical example of binding. So remember, protein binding involves reversible formation of relatively weak intermolecular forces between molecules. All right, so let's have an example. Let's say we've got some drug molecule. So for the sake of argument, we'll say that it's a phenyl ring that has an isopropyl group and an ethyl and a sulfonic acid on one end of it. And that's going to bind to some sort of a cavity or binding site of some type on a protein molecule. Now, when small molecules bind to proteins, usually they bind to sort of, caved in spots on the surface of the protein. Well, what does the surface of our protein molecule look like? Well, you'll recall from our discussion of protein structure, normally all along the exterior of the protein, you're going to have polar amino acids that stick out into water and interact with the water. I'm not going to draw the whole molecule, but I'll just draw, the relevant portion of it. But then you're also going to have some segments of your protein where every now and then you do have an exposed nonpolar amino acid. We'll say we have an isoleucine there, and we'll say we have a valine here, and we'll say we have an alanine there. But then there's also going to be spots where you may have, say, a lysine, serine, whatever. So how is this molecule going to bind to this spot on the protein? Well, what it's going to do is it's going to form favorable intermolecular forces. So probably what would happen is we would form something like this, where we have hydrophobic contacts between the hydrophobic part of our molecule and nonpolar amino acids on the exposed surface of our protein. And then we can have an ion pair interaction between the negative sulfonic acid group on our small molecule and the positive lysine on the protein. Now, this binding interaction isn't super strong. It's going to be reversible, right? But as long as the small molecule is bound, we've formed some favorable intermolecular forces. And therefore, the bound structure is going to be stable to some extent. Not super stable, because this is still reversible, but it's going to be stable. So the strength of binding, if we were to define the strength of binding, what do we mean by that? The strength of binding refers to the stability of the complex. So what do you think would influence the stability of the complex? Well, a more stable complex, all else being equal, is the one that has more favorable intermolecular forces. So the stronger these intermolecular forces are, the more stable our complex is, and the stronger our binding. So let's take a look at a way that we can express the strength of binding quantitatively. All right, we already said before that if you have stronger intermolecular forces in your complex, that means you're going to have stronger binding. So let's come up with a way that we can express this quantitatively. We're going to define our protein with a capital P. And whatever small molecule we're binding to it, we're going to call L. P stands for protein. L stands for ligand, which is just a general term for anything that your protein binds to. I don't like the use of the word ligand. To me, that's stolen from inorganic chemistry, and it means whatever is binding to your metal atom. But whatever. Again, we're stuck with the term, and I don't like it, but we have to use it. And then once our protein and our ligand bind, we have a complex, which we're going to represent as PL. And to keep things as simple as possible, we're going to assume that only one ligand binds to our protein. Okay, so let's think of our protein binding as an equilibrium. We're going to say that our protein and our ligand can bind reversibly to give us a protein ligand complex. Okay. Now, what we're interested in doing is figuring out what's the strength of this binding interaction. Well, you can imagine that if the binding interaction is pretty strong, that means the equilibrium would lie further over towards the right, towards products, correct? So let's define an equilibrium constant for this reaction. We're going to call it K sub A. A is for association, so we're going to call this association constant, not to be confused with Ka acidity constants. Ka is going to be equal to the concentration of complex divided by the product of the protein and ligand concentrations at equilibrium. Okay, so if you remember equilibrium constants, this should be pretty intuitive for you. If you have a high Ka, that implies strong binding. All right, but Kas are actually kind of inconvenient. There's a better way to describe the strength of our binding interaction. Here's what we're going to do. This may seem sort of counterintuitive, but let's look at the reverse of this equilibrium. So instead of looking at protein and ligand binding, let's start out with our complex and let's look at dissociation of the complex. So the equilibrium constant for this equilibrium, we're going to call Kd. Kd is also called the dissociation constant. And what's Kd? Kd is equal to the concentration of protein times the concentration of ligand over the complex concentration at equilibrium. All right, Kd is a term that you're going to want to know. This is how we would define it, all right? But it's not a very intuitive way to imagine it, because after all, we're looking at a dissociation, but we want to talk about the strength of binding. And, you know, counterintuitively, a smaller Kd implies stronger binding. But here's why we like KDs. Let's imagine that we're in a situation where the concentration of protein, free protein, is equal to the concentration of complex. What does that mean? Well, when that situation exists, if we have one ligand binding site per protein, which we're assuming that we do, then this means that 1/2 of all possible binding sites are occupied. So if you take these numbers and plug them into your expression for Kd, what happens? If 1/2 of binding sites are occupied, then that means that this term and this term are equal, so they're going to cancel, and the Kd is equal to the concentration of our free ligand. So our Kd is equal to the concentration of free ligand that results in 1/2 of protein molecules being bound to ligand. That's how you should think of Kd. And so you'll notice here, if this is a very small number, what is that implying? That means that even at very low concentrations of ligand, you already have half of your protein bound. Now, I want to point out something that's a common misconception. Okay, this is not the same thing as the concentration of ligand at which 1/2 of the ligand is bound to protein. That is not what KD is. The reason that is not what KD is that this model assumes that the concentration of ligand is very much greater than the concentration of protein. If the concentration of ligand is not very much greater than the concentration of protein, then KD tells you very little. So we're going to always assume that we have much more L than we do P. Now there's a way that we can sort of graphically see the relationship between KD and the strength of binding. So we're going to define a parameter. We're going to call it theta, and we're going to call theta the fractional saturation. What is this? Well, theta is equal to the concentration of protein that is bound to L over total protein, which if we have just one ligand binding per protein, it would be this, PL over PL plus P. However, if you have multiple binding sites, and they're all equivalent, that's a pretty big if, but if you have multiple binding sites and they're all equivalent, then theta is equal to the concentration of bound binding sites over the concentration of total binding sites. These are all equivalent. This is what you would have if you have just one-to-one binding, and this is if you have multiple binding. Anyway, without deriving it, I'm going to just state that it can be shown that our fractional saturation theta can be written as follows. Concentration of free L over the concentration of L plus KD. What is this telling us? Well, notice that theta, it's a fraction. It ranges from 0 to 1. Theta equals 0 means nothing is bound to your protein. Every single one of your molecules of protein is going to be free. And then 1 means every single last molecule of protein has L bound to it. Well, this equation is very handy because this equation describes a hyperbola. So if you were to make a graph of theta as a function of ligand concentration, you would get a graph that looks like this. So what do we see here? We see that as you increase the concentration of your ligand, you get closer and closer and closer to theta equals 1, which means that you're getting closer and closer and closer to every molecule of protein having L bound to it. So this is referred to as saturation. Saturation is the situation when your theta is approaching 1. But more interestingly, what else do we notice? What we notice here is that if you follow the y-axis down to theta is equal to 0.5, and then go down to the x-axis, the concentration of L at that point is going to be equal to your KD, your dissociation constant. So once again, KD is the concentration of L that gives 1/2 of protein molecules bound to L. A lower value implies stronger binding. So if your KD is 1 micromolar, that means that at a 1 micromolar concentration of your ligand, you've already got half of your proteins bound. Whereas if your KD was 100 micromolar, that means you would have to increase your concentration all up to 100 micromolar before half your proteins would be bound, because the binding is so much weaker. If you prefer thermodynamics, there's a final relationship that I can give you. The delta G, the standard delta G for binding, is equal to RT natural log of KD. R is the gas constant, T is the temperature. If you have a low KD, then that's going to give you a more negative natural log, which is going to give you a lower delta G for binding, and therefore a stronger binding interaction. Once again, everything that we've done on this slide assumes two things. We are assuming, first of all, that our ligand concentration is much greater than our protein concentration. And secondly, we are assuming that we have only 1-to-1 binding, or we have multiple equivalent binding sites. Now let's look at a different situation. We're going to talk about what's called cooperative binding. So I'm just going to tell you what cooperative binding is right out of the gate. Cooperative binding is an interesting phenomenon that you get whenever you have multiple binding sites and a KD that depends on how many ligand are already bound. If you don't have both of these, you cannot have cooperative binding. So here's the situation. This is how it works. Let's say that we have a protein that has 4 binding sites. It doesn't have to be 4. It could be anything 2 or more. But this was worked out for hemoglobin, which has 4 binding sites for oxygen. And so we are going to assume that we have a protein that behaves like hemoglobin. So you can imagine that we have our ligand and we have our binding site, but you'll notice that our ligand is round and our binding site is kind of square. So our protein doesn't bind our ligand very well. So we've got a relatively high KD for binding, at least at first. So a ligand binds, but it binds kind of weakly. The equilibrium, it kind of favors dissociation. But let's say that we end up in a situation where once we have a certain number of ligand bound, so once we have a certain amount of L bound, our structure of the binding site changes slightly. So you'll notice the way I drew this is that our binding sites go from being square to being round. So at this point, binding becomes much easier, or more precisely, binding becomes stronger. So that now our equilibrium favors binding. Now we have a much lower KD. So in other words, what happens is that binding, a certain amount of binding, leads to a conformational change, which causes a change in the KD, usually going from high to low. So another way of thinking of cooperative binding is that binding gets stronger the more L is already bound. Now, you may be wondering, does it ever go the other way? Does it ever start off binding very strong and then gradually get weaker? And the answer is yes, that does exist. It's called negative cooperativity, but it's pretty rare, so we're not going to talk about it in this class. Now, notice the way I drew this. It's not like once something binds, the structure of that binding site changes, and then when the next one binds, the structure of that binding site changes. It doesn't work that way. Notice how all binding sites change simultaneously. Right? In these structures, we have 4 square binding sites. And in these structures, we have 4 round binding sites. We don't have intermediate structures where you have, 2 round and 2 square or anything like that. So in most cooperative proteins, not every last one of them, but most of them, this is the kind of situation that you see. We have 2 structures of our protein, 2 closely related conformations. We have a weak binding structure, and the symbol for that is capital T. We call it the T structure. And we also have a strong binding structure, and the symbol for that is capital R. So usually we start off in the T structure, and then as more and more of our ligand binds, we find ourselves in the R structure. The T structure has a high KD, the R structure has a low KD. So now my question is, what does the theta versus L curve look like whenever you have a cooperative binding protein? What happens is when you have a cooperative binding protein, you end up with an S-shaped curve. It's not hyperbolic like it is for a one-to-one binding protein. So for example, in hemoglobin, on the x-axis, we have concentration of oxygen. Well, partial pressure of oxygen, but who cares? That's the same thing as concentration, basically. And we've got theta on the y-axis. What we observe is that our binding curve has this sort of S shape. It still saturates when you get up to very high concentrations, but what's actually going on here? Why does it have this kind of S shape? Well, the reason that it's an S shape is that it's a superposition, sort of a weighted average of 2 regular hyperbolic curves. When you start out binding, you're at very low concentrations of your ligand. You're in the T structure. This is the theta versus L curve for the T structure. And notice that it's going to have a very high KD. And then this curve, which is so steep that you can barely see it, this is the theta versus L concentration curve for the R structure. And its KD, it's so close to 0 you can't even really see it. And so what happens is, as more and more of our ligand binds, we start off in the T structure and then gradually switch over somewhere in between to the R structure, so that at very low concentration of ligand, you have mostly T, and therefore we are on the T curve. And then at high, we have mostly R, and then we switch over to the R curve. And in between the two, we're going to have a mixed population. Some molecules are in T and some are in R, so we're going to be somewhere in between the two curves. So this is cooperative binding, and cooperative binding is very important in biology. This has a big biological advantage. Why do you suppose cooperative binding is so important in biology? Well, it ends up resulting in a large change in the fraction bound as a function of a small change in your ligand concentration. So much of biology just boils down to proteins binding onto small molecules. And once the protein binds to that small molecule, its function changes in some way. And you'll notice that in the case of cooperative binding proteins, you get a big change in the amount of protein bound with a small change in ligand concentration, right? So for the case of hemoglobin, if you're breathing normal air, the pressure of oxygen in your lungs is way out here, and your hemoglobin is going to be essentially saturated with oxygen. But then when you go out into the tissues where the concentration of oxygen is much less, right, you need the hemoglobin to let go of that oxygen so that your tissues can be fed with it. Now, if we just had a regular non-cooperative binding protein, like the R state curve, what would you notice? Well, the difference in oxygen concentration between the lungs and the tissues is pretty significant, but the difference in theta is essentially nothing. Your theta would barely change. But because we have a cooperative binding protein, when the hemoglobin gets out into your tissues where there's not as much oxygen, the theta can drop pretty substantially. And that means that we're able to release our oxygen. This would not be possible with a non-cooperative oxygen binding protein, like myoglobin, for instance. So I refer you to the textbook. The textbook has an entire chapter on cooperative binding, chapter 9. It goes into far more detail than I care you know about, so it's okay to just skim it, but just get the concepts from it. However, understanding cooperative binding is going to be very, very important going forward in this course when we start talking about allostery and regulation of proteins in future chapters. I'll see you guys next time.